Alrighty, so we're going to get started. Thanks everyone for attending, in person especially. Today we have a special seminar from Philippe Boileau, who's a new member of CORE. His background, he's biostatistician, trained, PhD from UC Berkeley. He also did some work in industry, so he brings sort of that special perspective, very contemporary perspective. And his expertise, well, I'll probably mangle this, but it's investigating high dimensional data using machine learning and other biostatistical methods to identify potential heterogeneity of effect. And so he's going to tell us what he does. And hopefully, if you could benefit from his expertise, reach out to him. That's one of the purposes of this opportunity. Yeah.
Thank you. Before we get to our speaker, we just wanted to say that Phil, it's an honor to welcome you as our clinical trial methodologist as part of the ACT initiative. We're really happy to have you here. I think you're a start to what's going to be a very fruitful methodological hub here at the Institute. We're very happy to hear from you today, and welcome.
All right. So my talk is on finding treatment effect modifiers in clinical trial data, particularly high-dimensional clinical trial data. But before I get into the details of that work, I'll give a little bit of a background on myself and what Ben was talking about before. So high-dimensional analyses, assumption, lean inference, that kind of stuff, which is admittedly very jargony. So my research agenda is broadly interested in developing and applying these assumption lean methods, in particular to the quantification of heterogeneity of treatment effects. So that could be identifying effect modifiers, subgroup discovery, or quantifying variability in clinical trials, as well as designing clinical trials by defining new estimands, another jargony word that I'll define in a slide or two, generating or incorporating real-world evidence into the design of clinical trials, or even using real-world data from administrative claims or EMRs. So what is assumption lean inference? Well, it's a combination of two subfields of statistics, causal inference, which is nothing more than a framework for augmenting associational relationships with causal interpretations, and then machine learning, which is a field that's really dedicated to the development and application of algorithms that learn from complex data. And so when you combine the two, you get assumption lean inference, which is a set of methods that avoid making assumptions or convenience assumptions about the data to provide reliable statistical inference and potentially causal inference through a principled use of machine learning. And so what's really nice about assumption lean inference is it provides you with a roadmap that you could generally apply to any one. There's 4 general steps. And the first is simply defining the scientific question. So as a statistician, this is done with collaborators in medicine or in other fields. From there, you then translate this question into an estimand, which is just a statistical parameter that quantifies that question. And then from there, you can move on to the modeling stage, which is defining a suitable model that know about how the data was actually generated. And the pointers here, and like you'll notice that these arrows are bidirectional because it's not an even flow from step 1 to step 3. There's going to be a lot of conversations going on between collaborators to figure out exactly what the estimand is based off of the scientific question and how the data is generated. But once step 1, 2, and 3 are all set, you move on to step 4, which is simply performing statistical inference within that model of your specified estimand. And then from there, you get to interpret the results to hopefully answer your scientific question. So broadly applicable to a ton of problems. And this is in fact also recommended by the ICH, or this stands for the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. Like a real mouthful. But basically this is the recommendation by a lot of regulators in how to approach quantitative problems in clinical trial design. Now this contrasts with the traditional roadmap for statistics, which involves the same step 1, so defining your scientific question, but then going directly to the modeling stage where you're going to select the model based off of the outcome type, and then moving on from there to identify the estimand based off of this model. So you define the model, and then from here you're stuck with whatever estimand is stuck in that model. And then from here, you perform your statistical inference and then hopefully interpret a result to make sense of your question. Now, this is a good approach given constraints on computation, on complexity of the analyses. And so we've been using it as statisticians for 100 years, right? But there are some issues. So namely, there we go. So there are some issues, and here's an example that frequently comes up in clinical trials. So the question is, does a new therapy improve overall survival relative to a placebo? Now, this is an overall survival question, time to event outcomes. You might immediately jump to a Cox proportional hazards model. And if you're in a Cox proportional hazards model, then your estimate in step 3 becomes the hazard ratio. From here, you could perform inference by estimating the hazard ratio of therapy A against your placebo, possibly adjusting for prognostic factors, right? An issue arises here when the proportional hazard assumption of the proportional hazards model is violated. And in fact, this is often a violated assumption when you're comparing a new therapy against a placebo because the hazards are not proportional over time. And what that means is that it's quite hard to jump from step 4 into step 1 and actually make an interpretation. If you instead use the assumption lean roadmap, you'll define the same question, you'll move on to step 2, and you might say that given hazards are not proportional, we're going to use a different estimand, a different target parameter. We'll instead use the difference of the restricted mean survival time, which I'll show you in the next slide. From here, you'll model this outcome, given what you know about the data, which might be very little, especially if this is a first-in-human trial or if this is a phase 2 trial with relatively little evidence. You'll then perform statistical inference of this estimand using machine learning methods, and then be able to interpret this result in terms of your original question, hopefully without violating any assumptions. Potentially, it depends. Like it always depends on the question and what assumptions you're willing to make. And in some cases, it might actually make sense to use logistic regression. And this isn't to say that this approach is bad, right? It's just there are more assumptions that you have to make, which could possibly be violated, and therefore make interpretations hard or inference hard. And so this is kind of like a more robust approach. So this is the RMST for those who are wondering. This is the survival curve. And so the RMST is simply the area under the survival curve up until a restricted time point. So in this case, over here, you have an RMST of about 12 years, assuming the restriction is at 15 years. And so when you want to compare your group 1 versus group 2, so your new novel treatment versus your placebo, you could compare the area under these curves to get this, which is interpretable in terms of years. So here it's about a 2-year difference. So broadly interpretable across different kinds of models. Any questions? Oh.
I mean, I guess I was thinking about the proportional hazards assumption and getting around that assumption and the different modeling approaches that I'm aware of, but none of them invoke, to my knowledge, the machine learning or even the causal inference step. So is this RMST specific to, is that tapping into these two things?
That's a great question. So if I go back over here, in the most flexible model where you don't want to make any assumptions, you could use machine learning methods. But if you have a good understanding of how, for example, the outcome and the treatment and the prognostic factors are related, say they follow a logistic model or some parametric model, then you could use those to estimate the RMST, and that's totally fine. So it's a very flexible approach that allows you to incorporate exactly what you know or think you know about the data generating process. But if you're not familiar, you're not sure about that, then you could use these flexible machine learning methods. Okay, so going back to my research agenda, today I'm going to be talking about treatment effect modifier discovery in clinical trials, barring any other questions. All right. Okay, so this was work that was done while I was at UC Berkeley and interning at Genentech with my supervisor, Sandrine Zdua, my unofficial supervisor, Mark Vanderlaan, collaborator, Nima Hijazi, and Ning Lei, my supervisor at Genentech. And so the motivation from Genentech's point of view was this. Basically, they have a drug development pipeline that goes from basic and preclinical research through phase 1, phase 2, and phase 3 trials, and on to the regulatory approval and launch of a product, assuming the phase 3 is a success. Now, focusing on these the different phases of the trials, what we know as clinician scientists, and I know as a biostatistician, is that the cumulative amount of resources required as you jump through this pipeline increases steadily, both in terms of actual money spent, but also in terms of patients that are used or invited or to participate in these trials. And so the worst possible outcome from a clinical perspective, from a financial perspective for these companies, is that you end up with positive phase 1 and phase 2 trial results, but you fail at phase 3, meaning you've wasted all of these resources. Or you're not wasted, but didn't use them as well as you could. And in fact, about 54% of late-stage trials have failed among about 630 that were identified in public and commercial databases between 1998 and 2008. And this is probably an undercount, given that a lot of pharma companies will not actually publish, not necessarily publish negative findings. And so as I was saying, these trials fail frequently. And this is bad because sponsors are wasting resources and patients are not participating in trials that could potentially improve their outcomes. And so understanding why these late-stage trials fails is really needed to mitigate the risk in future trials, but also try and salvage the failed trial data, maybe use it for the design of new trials. And so there's many reasons why this pipeline might break down in phase 3 trials. One of the reasons that we think is, or one of the culprits we believe, is the presence of heterogeneous subgroups. All right. So I have here a rough sketch of a phase 2 trial, a 2-arm design, a treatment arm, and a control arm, and patients are randomized to these 2 arms. Now, in the data, we see that there are 3 subgroups that are unknown or unaccounted for, either because these pretreatment covariates are not measured or they just weren't used in randomization. And so when you actually arrive at the end of the trial and you want to evaluate how patients did in each arm, you're going to compare the treatment effect, right? And so each one of these subgroups potentially has a different treatment effect. So we see in the left over here, these two groups benefit on average much more from receiving the treatment than receiving the control. Whereas in this small subgroup over here, patients who have this profile based on pretreatment covariate 1 actually have negative outcomes under the treatment and very good outcomes under the control. But you don't see any of these because you're not actually accounting for these subgroups. You only get the marginal effect, the average across all subgroups. And what you're going to see is that there is a slight benefit to administering the blue pill over the red pill, or the treatment over the control. So that's good. You move on to a phase 3 trial. Everybody's happy. But now, due to bad luck, you have the same subgroups, but there's a different number of patients in each one. They have the same overall treatment effect within subgroups based off of the pretreatment 1 and pretreatment 2 covariate profiles. But when you take the marginal effect, or estimate the marginal effect by averaging across all of these subgroups, you end up with a losing medication. Your treatment now does not show evidence of being better than the standard of care or the control. So why is this happening? Well, it's happening because pretreatment covariate 1 is a treatment effect modifier. So sketched out over here on the x-axis is the values of pretreatment covariate 1, and on the y-axis we have the predicted outcome for different values of that pretreatment covariate. And in blue we have the outcomes of patients in the treatment arm, and in red the outcomes of patients in the control arm. And what we see is that patients who have relatively small values of pretreatment covariate 1 will benefit more, have better outcomes when assigned to the treatment arm than the control arm. And the opposite is true for larger values of pretreatment covariate 1, meaning patients benefit from the control medication over the treatment. And so understanding treatment effect modifiers can really help mitigate the risk in drug development. So this would allow us, or getting a handle on them would allow us to refine our clinical trial design by targeting subpopulations that really benefit the most from the novel medication. And it could also help with drug target discovery. So we understand sometimes, if we're lucky, the biological mechanism of the therapy by understanding these treatment effect modifiers. And the good news is that many data are collected as part of modern clinical trials, at least in industry, which gives us an opportunity to identify potential treatment effect modifiers and incorporate this into our clinical trials. The difficulty in this, however, is that there are often more potential effect modifiers than there are patients. And so a lot of standard methods don't actually work in this setting. And so this leads us to the first paper that we worked on in this sphere, where we asked two questions. Do standard statistical methods allow us to identify treatment effect modifiers reliably in high-dimensional clinical trial data? And if not, can a reliable method be developed? And so the problem setting is as follows. We're going to assume there are n patients distributed according to some vector O, which contains W, the pretreatment covariates, of length P. So this could be your age, BMI, and tumor gene expression. We then have a random variable A, which is the treatment assignment, whether or not a patient is assigned to the treatment arm or the control arm. And then we have our outcome Y, which is continuous or binary. And this will be the assessment at the end of the trial. We're also going to assume that the data is high dimensional. So that means that N, the number of observations, is approximately the same size as the number of pretreatment covariates P, or indeed P is much larger than it. And so we're going to say that the primary estimate in this trial is the average treatment effect, which is this beast down here. So all this is saying is that the average treatment effect is defined as the predicted outcome of patients assigned to the treatment arm minus the predicted outcome of patients assigned to the control arm. Another way of writing this is to look at the predicted outcomes of patients assigned to the treatment arm given their pretreatment covariates minus a similar object, but for the control arm, and then averaging over all their pretreatment covariates to get the ATE. And what's inside these parentheses over here is called the conditional average treatment effect, which is simply the predicted difference in outcomes between arms given pretreatment covariates. And so you could use this to identify patients who benefit more or less from being in one arm or the other. And so in terms of traditional methods, that's actually a quite easy problem. We know that when n is larger than p, we could model these treatment effect modifiers using, for example, linear models. So this is nothing more than the predicted outcome given treatment assignment and our pretreatment covariates, modeled by an intercept term, a treatment effect coefficient, a prognostic score coefficients, and then an interaction term between our covariates and our treatment assignment. We could also get a little bit fancier with things and look at the Kate modeling with a under linear assumptions. For here we have beta 1, which corresponds to the treatment effect or the ATE, and beta 3 is again an interaction term between the treatment and the covariates. And so what we're really interested then is quantifying beta 3s over here, because any beta 3 that is non-zero corresponds to a treatment effect modifier. And we could use traditional hypothesis testing procedures to get a handle on beta 3, to get p-values. When we're in the high-dimensional setting, so again, when we have more pretreatment covariates or more potential effect modifiers to look at than there are patients, we run into issues. We can't actually use traditional approaches anymore. We have to rely on p-analyzed methods, like, for example, the lasso, which you may have heard of, which simply shrinks these beta 3s down to a value of 0 when the algorithm believes that it's not important. Unfortunately, this approach does not allow for traditional hypothesis testing procedures. Inference is tough. What you have to do instead is rely on variable selection, reliable variable selection of these methods. And so the reliable variable selection of the lasso, for example, requires 3 key assumptions. One is sparsity, meaning that there are very few actual treatment effect modifiers. The second is negligible correlation, so that pretreatment covariates are relatively uncorrelated. And then 3, linearity. There's an actual linear relationship between your outcome, your exposure, or your treatment, and your pretreatment covariates. And as soon as you violate any of these assumptions, you kind of lose control over the reliable selection. That means you get increased false positives, increased false negatives, and ultimately that means that you can't really use this to de-risk your drug development pipeline. And I'll note here, actually, I'll emphasize that I'm interested in using gene expression, transcriptomic data, as potential treatment effect modifiers. And so genes are generally correlated. They're not necessarily linear, depending on how you transform them when you analyze them. And in terms of sparsity, well, if one gene influences an outcome and it's part of a larger gene set, then you could argue that all of those genes in the gene set also probably influence the outcome. And so generally, these assumptions are not met in the setting that I'm interested in investigating. And so relying on the assumption lean inference roadmap, we came up with a estimand that can help us answer. And it's called a treatment effect modifier variable importance parameter. It's a mouthful, so we'll just call it a TEM-VIP. And this TEM-VIP is a causally interpretable parameter of effect modification in a non-parametric model where we're not making many assumptions. And so the TEM-VIP for a given pretreatment covariate Wj is given by this beast over here. So it's psi j equal to the covariance, the conditional average treatment effect, and the pretreatment covariate in the numerator, divided by the variance of that pretreatment covariate in the denominator. And we're going to define this psi, or a psi, for every single one of our pretreatment covariates. If this does not look familiar because your intro to stats class, was a while ago, don't worry about it. I have a drawing. So we're going to go back to our original sketch over here. We have our predicted outcomes on the y-axis, the jth pretreatment covariate on the x-axis, and we see that the outcomes interact with this pretreatment covariate depending on group assignment, a equal to 1 or equal to 0. From here, we're going to compute the difference between the outcome in the treatment arm versus the control arm to get our conditional average treatment effect, which is potentially a very nonlinear, complex relationship between our pretreatment covariate J and the predicted outcomes. Now, it turns out that mathematically, we're not actually able to estimate this complex function well in a non-parametric statistical model without making a lot of assumptions. What we can do, however, is try to approximate this complex line using, for example, the best linear approximation in green over here. And in general, this linear approximation will capture the trend in that line. And so our psi naught j, the TEM-VIP that I showed you before, is nothing more than the slope of this green line. So in this case, we have treatment effect modification because this Kate is not flat. There is some kind of relationship between our pretreatment covariate and the difference in predicted outcomes. If, for example, our pretreatment covariate is simply prognostic, but does not interact with treatment assignment, then our Kate is flat, and so too is our line. And so our slope is going to be equal to 0. So this will allow us to perform inference with a relatively simple, I think, interpretation for our treatment effect modifiers. So as I was saying, this psi j is a simple linear regression coefficient obtained by regressing our Kate on the jth pretreatment covariate. And its interpretation is as follows. A unit increase in Wj causes the treatment effect to change by approximately psi j. And again, we're going to have one of these psi j's or make a statement like this for every single one of our pretreatment covariates.
It's just so you can make comparisons between different pretreatment prognostic variables.
Basically, it's like a standardization factor. That's exactly it. That's a good question. But it seems complicated. There's a lot of math here. Just think about your simple linear regression coefficient, a slope, and that's all there is to it. And so now that we've defined our parameter, we could talk about performing inference about this parameter using an estimator. And so we derived a non-parametric estimator, which I don't have written here, which is probably better for all of you. But it depends on the predicted outcome given pretreatment covariates and the propensity score. And these components can be estimated using machine learning or, as Ben was talking about before, parametric models if we know for a fact that those relationships are parametric. Now, what's really nice about this setting in clinical trials is that propensity score is actually known. So this really simplifies the estimation procedure. And we end up with a guarantee that our estimator is unbiased in large samples, regardless of how complex the relationship is between our outcome and the pretreatment covariates. And so what that means is that we don't have to rely on sparsity or linearity or correlation to get a good control of our false positive rates. really simplifying the problem. And so I have time for this, yeah. So I'll just make a small note on linear models in the traditional and assumption lean framework. So we're starting with this potentially complex relationship between our outcome, our treatment, and our pretreatment covariates, right? And the traditional approaches make an assumption that this complex relationship is linear for convenience. And this is okay, because maybe it's convenience due to a lack of time to fit machine learning models or a lack of compute power to do it. From here, you then perform a statistical inference as if this complex relationship truly is linear, and then your approximation to reality will provide reliable evidence when that approximation is good enough. Of course, good enough is somewhat hard to quantify, but this is the general approach, right? In contrast, the assumption lean approach that we're using summarizes the complex relationship using a linear approximation. So we're accounting for the fact that this is an approximation. We're then performing statistical inference, again, accounting for this approximation. And then at the end, we get a principled approximation that provides reliable evidence for decision making. So maybe it's a subtle difference, but really what this allows you to do is to quantify uncertainty while accounting for what you don't actually know about the problem, which is whether or not relationships are linear. And so to see if this matters, this subtlety matters, we performed a simulation study comparing three methods, our TEM-VIP estimator, and then these two traditional estimators. So a penalized Kate and a lasso. We then considered three different data generating processes where we simulated 500 genes based off of actual gene expression data and assumed that about 20% of them were truly effect modifiers and the rest weren't. And we also assumed different relationships between the outcome and these effect modifiers. So the kinked treatment effect modifiers are somewhat nonlinear, they're piecewise linear. This linear model over here assumes that the relationship is linear, which is good for the traditional approaches. And then we have a very nonlinear relationship between our outcomes and our pretreatment covariates on the right. And so the first thing we did is measure whether or not we could actually control our false positive rate with our method. And this false positive rate is that red dotted line over here set at 5%. And what we find is that our method is the only approach that can control this false positive rate at sample sizes starting at about 250, going up to 500. And even at a sample size of 125, which is like a relatively small phase 2 trial, you still have pretty good control in some settings. Now, it's not enough to just look at whether or not you can control false positive rates. You also want to look at how often you're correctly classifying non-treatment effect modifiers and treatment effect modifiers. And so we looked at the true negative rate. So how well are we doing at really classifying non-treatment effect modifiers as non-treatment effect modifiers? And we see that our yellow line is near the 100% mark. So in most cases, we're doing a good job of identifying non-treatment effect modifiers. The other methods, on the other hand, sometimes actually decrease in performance as sample size grows, owing to model misspecification. So when you make the assumption that things are linear, but they're not, like in this case over here, some of these methods are going to suffer and actually get worse over time. Bad news for reliable inference. And finally, the big question is, are we obtaining good false discovery rate control because we're very, very stingy when it comes to identifying the true effect modifiers? And the answer is kind of, but not really. In small sample sizes, we're not doing a great job of identifying all these treatment effect modifiers. The ones that we are identifying are actual treatment effect modifiers, which is good, but we're missing out on a lot, right? Only like maybe picking up a few percent out of the total. As sample size grows, however, we see that our method performs somewhere in between this dark blue method, the penalized Kate, and the lasso. And so what this says is that you could get reliable treatment effect modifier discovery in terms of false positive rates, while also getting a pretty good job of identifying all the true effect modifiers, probably based on the size of the effect. With those simulation studies in hand, we were quite happy with what we were seeing, so we decided to apply it to Emotion 150, which is a phase 2 trial in metastatic renal cell carcinoma that Genentech ran a few years back. And so Emotion 150 wanted to compare whether ICIs in combination with VEGF inhibitors improved outcomes versus VEGF inhibitors alone. And so the patients in question are actually considered two arms from this trial of MRCC patients. Those from the VEGF inhibitor arm, and those are the ICI plus VEGF inhibitor arm, for a total sample size of about 150. We then took the 500 most variable log transform genes from these patients, it's taken from their tumor samples, and we treated those as potential effect modifiers. And our outcome here was the objective response. So a binary outcome of whether or not they partially or completely responded to treatment. And our findings were that we identified 92 genes using a 5% alpha level that seemed to modify the effect of the ICIs and VEGF on outcomes. And what was really nice about this and what got the scientists, the clinician scientists at Genentech excited was that nivolumab had a trial running in the same indication. Well, I actually don't have this written here. Nivolumab had a trial in the same indication that came out around the same time. It was published at the same time that we found these results. And these 92 genes overlap significantly with the treatment effect modifiers they found in that trial. So there's some biological validation. Weak, but still pretty good results. Any questions on this? I'm going to move on to another. Yeah, you want to pass the.
So I'm very interested in the modifying effect of sex, in trials. And more often than not, we have the forest plot and there are the subgroups. And, you have, for women, it's kind of very wide confidence interval because there's very few women who are included in trials. And then for men, it's much narrower. But like more often than not, like there's no effect modifying effect that's found in many, many of the trials. So, I mean, what's the likelihood that you really have heterogeneity enough, to really falsify or modify the results, given that many, many clinical studies have never found effect modifying. I mean, I think for sex, it's often because there was not enough women included. And conclude that there's no effect modification by sex, but there might be. It's a question of statistical power. So that's kind of one thing, how do you, what's your expectation to find effect modifying when like we rarely find them? Maybe we don't look at, enough potential factors. That's possible. Another question I had was, the idea of response versus non-response. Like, obviously, this is towards like precision, medicine or health. We want to make sure that we design or test drugs that will eventually, really be effective on the person themselves. So you want to identify the predictors of response, So it's kind of modifying the effect. But what about a model where, you have a trial and then you have responders versus non-responders? I mean, whether they were randomized to either, arm, and then you test those as potential effect modifier. You know what I mean? Like because at the end of the day, you want to give the drug to the person who responded based on some of those characteristics. So you will basically use these characteristics to prescribe, people who are most likely to respond.
Yep. That's a great question. Maybe I should go back over here. So what we're doing is we're finding effect I'll answer your second question, then I'll go back to the first. So what we're doing is finding the effect modifiers here, right? This helps us provide some insight as to whether or not there is treatment effect modification. We're not yet at the level where we could identify subgroups that benefit more or less from the treatment. That actually requires a next step using the treatment effect modifiers to try and get a better handle on this curve over here, which can then be used to say, hey, okay, like patient A, for example, ends up over in this section of the curve, so therefore they should receive control, whereas patient B is placed over here based off of their profile, so they should receive the treatment. And that's the end goal, but we're just trying to slowly build up to that in a way that's reliable. In terms of your first question, you're completely right. It's so dependent on the indication, on the potential effect modifiers, on whether or not you actually have a treatment effect. There's a lot of what ifs that go into this. And so the best thing you could do is try and run a simulation study before you actually run your trial with what you think you know about the problem and see whether or not this approach would actually pick up on treatment effect modifiers. And potentially in the trials that you're working on, like, they're underpowered or maybe there really isn't effect modification, but there might be if you had larger sample sizes. So it's
Could you go over some trials that showed that there was no effect modification by sex, and then use this method to actually find?
Potentially, like, right, so this is a tool for identifying treatment effect modification. Maybe other approaches are not able to do it, but then reapplying this method to those trials, it might find a significant effect modifier. That's possible, for sure. Yeah.
Dr. Easer would like to understand how there was not an overfitting issue for such a small sample.
That's a great question. So I had this complexity from the audience, but basically there are machine learning tools that you can use to avoid overfitting called cross-validation or cross-fitting. And so that's what's used under the hood here. So we're avoiding selecting basically very, very aggressive machine learning algorithms because they won't generalize, especially in smaller samples, automatically under the hood. Yeah. Okay. So we were pleased with those results. We felt like we had a successful pilot method, if you will. But one of the issues is that the TEM-VIP approach we described is only appropriate for the average treatment effect, right? Now, that's not the only estimate that we're interested in looking at in clinical trials. So for example, you might have oncology trials that are interested in survival, so time to event outcomes, which you might quantify using some function of the restricted mean survival times, which I showed you before. Or if we're in a multiple sclerosis trial, we might be interested in reporting the relative risk of the number of events that happen over a fixed period of time. And so in all these cases, the TEM-VIP for the AT is not appropriate. It's very parameter specific. And so we wanted to develop a framework for identifying, creating, and estimating these TEM-VIPs for other estimates. And so what we came up with is a plug-and-play algorithm that allows you to create these TEM-VIPs for bespoke estimates in parallel study designs. What's really nice about this algorithm is that it also provides instructions for obtaining causal machine learning estimators of these new TEM-VIPs in a very straightforward way, and that these estimators have desirable properties like the ones I showed you with the ATE. Meaning in a clinical trial, when you apply them, you could really relax assumptions you make about whether, the outcome is a relationship to pretreatment covariance is truly sparse or linear. And so we worked through this algorithm and provided some examples. So we provided the estimates for the ATE like we had in the past, but also for the relative risk, the RMST difference, and the RMST ratio. And all these estimates are useful in different outcome types, ranging from binary counting continuous to time to event. And so again, we want to apply this new algorithm to a data set to see if there was any historical validation of our approach. And so we did that. We performed a reanalysis of the FINHER clinical trial data. So the FINHER clinical trial data was a trial of HER2 positive early stage breast cancer patients who were randomized to receive trastuzumab with or without, or a placebo in addition to the standard of care chemotherapy as an adjuvant treatment. The reason that we're interested in investigating treatment effect modification in this setting is that trastuzumab is a targeted therapy for HER2 positive patients, but actually plenty of biomarker positive patients actually have a resistance to this therapy. And so what we did was took the 500 most variable log transform genes of the 201 patients in this trial with gene expression data and ran it through our TEM-VIP for disease-free survival. And our findings were that there are actually numerous genes linked to trastuzumab resistance, and that most of these genes were actually already documented in the literature, or at least the most significant ones were. So this is what the actual results look like. So there's a lot going on. I'll walk through the panes one by one. From the first pane, we have the distribution of p-values and the cumulative, the distribution of nominal p-values by the cumulative distribution of p-values. And if there was no treatment effect modification, this black line over here should fall on the dotted line, but it doesn't. So this indicates that there's evidence of treatment effect modification. Of course, this is not enough to say that we have true treatment effect modifiers without looking at whether or not something is clinically relevant. And so it's hard to see over here, but our TEM-VIP estimator for the disease-free survival has or designates a decrease of 0.05 as equivalent to a change or a decrease in 18 days of disease-free survival. So for each unit increase of gene expression, yes, for each unit increase in gene expression, you have a change in the treatment effect of 0.05, which leads to a change in 18-day disease-free survival. And when we apply our p-value cutoff as well on the y-axis over here, we find that all these red genes are both clinically and statistically significant. We then take these genes and put them in this heat map over here. So on the y-axis or the columns, we have our predicted treatment effect modifiers. Again, the genes in red over here. And on the on the y-axis here, this is the x-axis. On the y-axis, we have our actual gene expression data for every individual patient. And then the cells represent gene expression that are log transform in a range of 1 to about 11 or 12. And what you see is that there at least seems to be a presence of subgroups based off the fact that you have patients with very distinct genetic profiles. So for example, this over here is a subgroup. This over here is another subgroup, as is this chunk over here. And so while we stopped short of actually investigating whether or not these subgroups had differential or heterogeneous treatment effects, there's a good chance that there is, simply because we identified them using treatment effect. So that's kind of like the next stage or a future paper to investigate. Any questions? Yeah.
Like the TEM-VIPs that you're estimating, that the gene expression levels that are associated with different treatment effect cluster within an individual. I guess that says that they're along some same biological pathway.
Exactly. That's what's nice over here. So for example, if you look at this group of treatment effect modifiers over here, you might want to investigate to see if they truly are on the same biological pathway. Same thing with this cluster of genes over here, 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 and here. So what's nice about this actually, and thanks for bringing this up, is that it's actually easier to interpret these treatment effect modifiers and you get the whole pathway or the genes, which is what this method provides. Whereas traditional approaches like the lasso would only spit out, for example, a single gene from a correlated set, just based on how the math works under the hood. And so you end up with a pretty interpretable way to create subgroups. Any other questions? Are these used now? I'm not aware of panels that are using like the genes that we find here, like in this particular corner over here, that are highly significant and have a huge effect, but maybe it's in the works. It's something that you could do based off of this research though, or like try and get going.
Really interesting everything that you're telling us today. So for this reanalysis, for example, for that specific clinical trial, you used pre-identified genes that you knew that they had some biological effect with the outcome, or you used genome-wide data, so searching everything across the genome, or you had some because the clusters there are really
right, great
impressive.
Great question. Thank you for bringing that up. I just took the most variable genes, the 500 most variable genes. That's it. I didn't go and talk to clinician scientists and ask them what genes should be included. If I was smart, I would have. I should have. If I worked with you guys on this, I would. But this was purely like agnostic to like prior evidence to see if we could actually pick up what other studies have already identified in the literature. And so, no, this is purely based identified through data adaptively, if you will. But that's an interesting point, again, if you're collaborating with scientists, what's nice about this methodology is that you can enforce, you could say Phil, I absolutely want gene X to be included in my final set of treatment effect modifiers based off of evidence that I have before I actually define my subgroups and go on to, create a gene panel and recruit in a new trial based off of that gene panel. That's something that can be done, right? It's just not what we did here. So there's a lot of ways to get clinician input.
Data that you need to go back and reanalyze that clinical trial that was already published is really what was collected already. So yes, we were using the clinical features or the genetic features or laboratory values that were already part of the initial trial.
Exactly. Exactly. So basically, this is actually taken from this data set was taken online. It's openly accessible, and it provides transcriptomic data, it provides outcome information, it provides HER2 positive status, what medication the patients received. Since publishing this paper, I think Genentech found out and actually took away the outcome variable, so you can't repeat this analysis, unfortunately. Or no, they took out the HER2 positive indicator, so you can't actually repeat this analysis, which is unfortunate. But yeah, you could just take historical data, run this approach on it, and get results.
Like very recent and like landmark clinical trials that for the first time in type 1 diabetes, we have something else to use rather than insulin. And it's a monoclonal antibody across, like the lines of, like along the lines of the medications that are being used here. And the outcome was very similar to what you were describing, that we couldn't identify, like a clear effect of that novel medication, likely because there were certain modifiers that were not accounted for. So we're going back and sort of reanalyze the data could open very novel pathways. So very interesting. So maybe I'm going to email you with
Thank you.
if you're interested in a collaboration.
Yeah, I'd love to. Actually, I'm going to talk about this right now, actually. So I have like future work in terms of methodology, but also in terms of applications. And the first application that I was really thinking of is the reanalysis of historical clinical trial data. And so this would allow you really to just basically, if it were to work, right? And you were to identify treatment effect modifiers, then you can inform the design of follow-up trials. That's an option. You could generate hypotheses to be tested in new studies, whether that's at the bench or in new trials, or just, perform a post-mortem, understand why the trial didn't work out, and it costs you effectively nothing other than compute and a stat person stat, a data set. I think another interesting application is to phase 4 in pharmacoepi trials. So I've been presenting this method in the context of clinical trials that are randomized, but because we're relying on machine learning methods, there's actually nothing stopping us from using this in observational data as well. And so I think it's, given that there's so much observational data available, it could be a really interesting source to mine to either, again, inform new trial design, generate new hypotheses, or even just understand why there's a difference between phase 3 trial results in terms of safety and tolerability versus what we're actually seeing in the real world post-market in a phase 4 trial. In terms of methodology now, I think one big issue with this method is that it's really only appropriate for parallel group designs. But we know that there are way more complicated designs out there that are needed to answer interesting questions. And so what I'd like to do in the future is extend this approach to longitudinal designs with repeated measures or even time-varying treatment. And I think this would really broaden the applicability of the approach and again, allow people to reliably identify treatment effect modifiers in more settings. Now, another thing that I think is interesting is to actually use these TEM-VIPs. I alluded to this before, is using our TEM-VIPs to identify subgroups. So once I get this, or we get this heat map, now I'm kind of just saying, so what? We have clustering, that's cool. Maybe the genes make sense. But let's actually look into these groups that we're seeing and determine whether or not there is heterogeneity of treatment effect. Do these groups experience the same outcomes across the board? If not, then that's even more of a reason to go after new trial designs or new trials to investigate treatment effect heterogeneity. This is challenging because you're going to be reusing the data, and so you could run into issues of overfitting. But again, you could rely on some pretty novel techniques to try and avoid this as much as possible.
We have someone in the chat who has raised their hand.
All right.
Ruth, would you like to ask your question?
Yes, please, if I may. Thank you for a very interesting presentation. One of the things I was wondering about is the issue of adherence when we're talking about real world data. We don't necessarily capture this, but nevertheless, there could be patterns based on patient characteristics where we could represent patterns in adherence or non-adherence rather. Any thoughts about how to incorporate this aspect?
Yeah, that is a great question. And I'll actually say that the TEM-VIPs for disease-free survival that I showed you accounts for informative censoring, for example, or at least can account for it if you wish to model that. But this all boils down to, I'll get there eventually, to this framework over here. So the easiest thing to do, like when you're modeling your scientific question, is to assume that there's no dropout, there's no informative censoring, there's no issue with adherence. But if there is, and you know it could affect results, and you're going to want to incorporate it over here. And once it's incorporated over here, then you'll be able to account for it in your computation stage where you're performing actual statistical inference. Again, potentially using suitable machine learning methods. And that's actually a really interesting problem. There's not much work there's not enough work being done on it right now, partially because it's very hard on the math side of things, but it's definitely work worth doing.
May I ask a follow-up question?
Of course.
On handling missing data, again, I realize you were talking clinical trials, but many times, again, large administrative databases that one could use to pursue this kind of work have periods where data might be missing with regards to changes in prescriptions and so on. Tools to address those would also be invaluable.
100%. And one of the issues, unfortunately, is that like some of these administrative sources of data or real world data are just too poor to actually glean any insight from. And so unfortunately, like this approach only takes you so far. If your data is really garbage, it'll only spit out garbage results. There's no magic wand to fix that. But there are slight fixes or tweaks that you can make to this design to try and overcome to an extent these issues. This is a really great question and a really great asterisk, I think, on these machine learning based approaches. They're not fix all, cure all. You know, they do have limitations like this.
One question that came to my mind is that you've transferred assumption to approximation. So have you tested, the error of your approximation?
We haven't. What we did though is think about when our approximation fails, right? When our approximation is going to fail to identify a true treatment effect modifier, simply because this linear approximation is incorrect, right? And so this line might be flat like this when you have a perfectly parabolic conditional average treatment effect. So this triple line would be like a U or an N, in which case this line would just give you something flat and indicate that there's no treatment effect, and you would miss it. In terms of stats, the parameter is correctly specified. We're doing a good job estimating that parameter. So the real issue is with the actual parameter, not with the underlying methodology. What you can do to overcome that is transform your pretreatment covariance. So if you have that parabolic shape, you could just consider squaring, taking a log transform of your variable, and then running that through this algorithm to identify whether or not it's a treatment effect modifier, but on a different scale. But that requires some prior knowledge and some work. So yeah, that's a good question. Does that kind of answer it, or? Yeah. Mm-hmm. Yeah. Yeah.
One thing that's interesting is the application of phase 3 to phase 4. And, often in trials, you have very few, you have clinical variables, but we always are concerned that we have, maybe sometimes, sex is there, but ethnicity might be there, but very few psychosocial variables are there. So it's very important, to link, trial data to what's going on out there and identify the potential modifiers, in a more like a psychosocial, so that, leaning, towards equity of application of treatments, to the people who are really responding.
That is a great point, right? So what you're saying is that if you see that like sociodemographic characteristics in the trial are different from sociodemographic characteristics of the patients in the real world, and you see that the outcomes are different than what the trial would suggest, then potentially you could identify whether or not those sociodemographic characteristics are effect modifiers. Right.
Yeah, but the problem, you don't have them in most trials.
Mhm.
So you might find a different effect in phase 4.
Gotcha.
Okay, but by having data on the psychosocial demographic variables, you may actually correct or explain why it's different from phase 3.
That's a great point.
Yeah, so it's a kind of a psychosocial modifier, if you like.
Yeah, and honestly,
in the real world,
great motivation for actually including or trying to collect these as part of clinical trials, right? Yeah. All right. Well, thank you.
Okay, well, thank you so much, Phil, for a great presentation. We now have an hour for meet and greets. We welcome you to come speak with our presenter, discuss your ideas, and hopefully come up with possibilities for collaboration.
So my office is in B245. I'm here on Thursdays and Fridays. And so if you want to come and chat about this, just drop on by. I'm happy to talk.
Thank you.